Hello. Good morning, everyone, and thank you for joining us bright and early for this year's ASN presidential address. My name is Becky Fuller, and I'm president-elect of the American Society of Naturalists, and our speaker today, Dr. Dan Bolnick, hardly needs an introduction, but I'm going to give him one anyway. Dan is a professor of ecology and evolutionary biology at the University of Connecticut and one of the most influential evolutionary ecologists of his generation. His work has reshaped how we think about individual variation, not just as background noise, but as a fundamental driving force in ecological and evolutionary processes. His work combines empirical fieldwork with mathematical models and genomics to examine how host-parasite coevolution occurs, immune system dynamics across various populations, and ecological speciation in three-spine stickleback. I think many of us have known Dan for quite some time. Dan earned his PhD and completed a postdoc at the University of California, Davis. He was a faculty member at the University of Texas for several years before returning to his beloved New England and joining the faculty at the University of Connecticut. He's the recipient of numerous awards, including many of the awards that we give out here at this meeting, including the ASN Young Investigator Award, the SSE Dubjansky Prize, the Ecological Society of America Mercer Award, and the ASN Distinguished Naturalist Award. He was also an HHMI Fellow, and if you try to go and count the number of papers and grants that he has, you're going to be counting for quite a while because the list is very long and impressive. And according to reliable sources during his graduate school days, he was known as Darwin II, which honestly checks out. Dan has long served the American Society of Naturalists in numerous roles, including as our secretary, as editor-in-chief for our flagship journal at the American Naturalist, and currently as president. And I really want to point out that in both his editorial roles and his presidential role, he has guided our community with clarity, principle, and just thoughtful leadership during some really challenging times. Today, he brings those same qualities to this address as we reflect on the crisis that we now face and the path forward. And so please join me in welcoming Dr. Dan Bolnick.
Thank you for that introduction, Becky. So, let me see if the clicker's working here. Just a quick rundown of what we have ahead of us. I'm going to start not with my address, but with some ASN business and some conference business. Tell you quickly about some ASN events that are happening the next couple of days, and then I'll get to the fun stuff. So quick overview of some meeting announcements. Just a reminder that there is a meeting code of conduct. I expect everybody has read it very carefully. And in all seriousness, you do need to abide by this. It is extremely important, and we take this extremely seriously in all of these societies. To help enforce this, there is a safety officer, Grand River Solutions. There's information about contacting them to get help with any incident that does arise. There are also EVOL allies. Various pins denote them, who have training in how to help respond and intervene in situations and can help guide you to contact the safety officer for follow-up. On to some ASN business. I'm going to give out some awards. There's a few awardees who are in the audience today and a number who are not. Those who are in the audience, if you can start to work your way up in just a moment, I have plaques to hand out, and I'll just ask that applause for the in-person awardees hold off. We'll bring all four of them up on stage, give plaques, and then we can give them a good round of applause. So the American Society of Naturalists, which I've been very, very honored to have a chance to interact with for many years, when I told my kids that I was going to be giving a presidential address and that I was elected president of ASN, my younger daughter paused and said, "What does that stand for?" And she thought for a minute and she said, "Oh, it must be the American Society of Nerds." So I just thought I had to share that because I really like that. I'm quite proud of it. So reveling in our joy of understanding and inquiry and interested in things in that role, I'm going to hand out a couple of student papers or announce a few papers. So first of all, The Student Paper Award is administered by the editors of the American Naturalist. This goes to an outstanding student paper in 2024, and this goes to Dr. Miller for her paper, Genetic and Phenotypic Consequences of Local Transitions Between Sexual and Parthenogenic Reproduction in the Wild, which was published last year. Dr. Miller is not here, nor is our honorable mention, I believe, Dr. Matthias Silstrom, for his paper, Anisogamy does not always promote the evolution of mating competition traits in males. Next, we have the Early Career Investigator Awards. They will be giving talks later today, this afternoon, so I believe they are all here. And so our Early Career Investigator Awards include, and if you can work your way down as I announce your name, I'll give you plaques and we can give you a round of applause. Dr. Jesse Williamson. Dr. Ashwini Ramesh. Dr. Liz Carlin. And Dr. Anna Doar. Are any of them in the audience yet this morning? Yes, I see a few people walking down. I think we have a few of them who are getting ready for their talks. All right, come on up, yeah. Okay, moving on, the next one is the President's Award. This is one that is my honor and obligation to give out. The president of ASN each year has to read the entirety of the American Naturalist from end to end the preceding year, so 2024, and pick an outstanding paper, and that turns out to be very hard because there are a lot of extremely good ones. And so I wrestled with this a long time and chose a paper by Dr. Lisa Bush titled Multitrophic Higher Order Interactions Modulate Species Persistence. She and her co-authors really did a tour de force, brute force experiment, very elegantly done, but an intense amount of work, creating all sorts of combinatorics of species interactions to ask how the presence of species A modifies the interaction between species B and C in a multi-species factorial approach complemented with some mathematical modeling. And it's really a spectacular paper. I highly recommend it. Then we have the Distinguished Naturalist Award, which goes to a mid-career scientist within, I think, give or take 20 years of completing their PhD. And this year's Distinguished Naturalist Award goes to Dr. Andy Suarez, who is an ant person through and through. And so he really has the passion for ants that really define, the passion for natural history that really defines this particular award. We're almost done with the awards. The next is the Distinguished Achievement Award. I think the full title is the Distinguished Achievement in Conceptual Unifications of Biological Sciences, but I think it's a bit of a mouthful. And this year's Distinguished Achievement Award goes to Dr. Stuart West, who has served this society extremely well over the years and published a large number of really hard-hitting conceptual papers. We also, as a society, have a modest number of honorary lifetime memberships. These are given to senior members of our community who have done exemplary service to the society and to the community through a combination of research and service. And this year, we're lucky to be able to give out two of these. One is to Dr. Joel Kingsolver, who has been editor-in-chief, president, a couple of other roles for the society, and of course has published a phenomenal number of papers that really have guided many of our thinking about phenotypic plasticity and the effect of phenotypic plasticity in evolution. And then the second distinguished honorary member is Dr. Doug Futuyama, who I think we probably all grew up reading his textbook, and really a phenomenal thinker in evolutionary ecology. When I think about presidential addresses for ASN, I think one of the defining ones is his presidential address from the mid-'80s or early '90s about whither the naturalist. What is it that defines a naturalist in this era? And the paper that he wrote after that talk really was a guiding star for me in my career. And so it's a real pleasure to be able to announce this one. So those are the awards. I have a couple of quick bits of society business to announce. First of all, send stuff to AMNAT. As a former editor-in-chief, I can tell you that we are one of the boutique journals that really, I'd say it's artisanal publishing. We really give each paper an intensive amount of attention. We care very deeply. There's a tradition at AMNAT of detailed, thoughtful, constructive review. In my 5 years as editor-in-chief, I felt like the quality of the reviews that I saw and the quality of the responses that I saw was really outstanding, and I was extremely proud of all of you, our community members, for the level of care, attention to detail, and thoughtfulness that went into the review and editorial process. And so I think AMNAT's a great place to send your work. I also want to highlight that AMNAT really strives to keep things affordable. It's a not-for-profit publisher, the University of Chicago Press. We have open access any way you want to do open access. We also have the ability to publish in more traditional format. It's a hybrid journal to ensure that publishing is financially available to anyone. We have fee waivers for people who don't have support. We also have been a leader in, I'd say, reproducible science, in making sure that not just data, our data archiving policy began 15 years ago, but then we introduced code archiving. We introduced an editorial team devoted to reviewing the archives of data and code to make sure that they're complete and reproducible, and I'm extremely proud of the quality of science that reflects and produces. A couple of events to remind you of. You're clearly here for the presidential address. I don't need to remind you of that. After this is the Vice Presidential Symposium. Amy Angert will be leading a group of colleagues talking about evolutionary ecology in human-altered conditions, and I think that's going to be an extremely interesting series of talks to listen to. There's an open forum for members of ASN. Simultaneously, there are open forums for SSE and SSB. I'm reluctant to mention those because you all really should be at the ASN one. This is a chance to come talk to the leadership of the society and learn what we can do for you, learn what you can do for us, learn how to help the society and how the society is helping you out, and give us feedback and talk to us about what we can do better, what we can do that might be new. After that, is the Early Career Investigator Talks, and that's going to be an outstanding series of lectures. It always is. And then on Sunday at lunch, there's the ASN Graduate Mixer. ASN faculty, please attend that. You're welcome. It's a great way for you to meet grad students and up-and-coming researchers in our field. And then there's the ASN Award poster award competition on Sunday night. So now's the point where I have to transition to my talk, and I'm going to have a sip of coffee to help me along. So there's a long tradition of ASN presidential addresses, and these often are about what it means to be a naturalist, like Doug Futuyama's talk, or how do species coexist, like Hutchinson's talk that became his Homage to Santa Rosalia paper. And so it was very tempting a year ago when I was sitting in the equivalent room watching Jeff Connor speak, thinking about what I would talk about. I wanted to have some inspiring science story. And I was thinking something along the lines of talking about how we go out with theory in mind to test theory, but we need to pay attention to the surprises, to the things that we're not looking for. And we might spot something like this tapeworm here that wasn't our reason for going out and doing a project, but it might turn into something even more interesting in the long run. So a story about paying attention to the things that we're not looking for. And I would weave in some helpful career advice to the early career scientists in the audience. I toss in a couple jokes or two to keep things lighthearted, because this is just after breakfast. Half of you are jet lagged. We need to keep this lighthearted. But then, you know, some things happened in Washington, D.C., so lighthearted might not quite be the right tone. So I decided eventually that I really couldn't give the talk that I wanted to, and I needed to talk about the elephant in the room. Since January of this year, it's become apparent that the trajectory of science in this country, which will influence science in many other countries as well, is changing and is at risk. And this will affect us all as researchers, professionally, and also individually as people, as members of the broader society and of scientific societies. And I was hesitant to pivot to a political talk because those of us in this room are united by our love of biology and biodiversity, understanding this, but we're not all in the same political party, we're not all of the same religious beliefs, we're not all of the same ethics, and I think it's important to remember that we are a diverse group of people in many ways. And so I don't think I can speak for everybody in this room on a political issue necessarily, or I don't trust myself to do so. And yet, political issues are affecting us all, and of course I'm talking about a variety of things here, but let's start with grant funding. The current NSF proposal to Congress, directed by the White House, calls for a 50% cut to itself. But that sounds bad, but if you look in the details, environmental biology and integrative organismal systems are looking about 90% cuts. And that would devastate this entire academic community. It would put at risk these conferences. NIH is looking at 40-ish% cuts. And even on top of those cuts, programs are getting archived and not revived. Grants are getting revoked. Funded grants, the money's not showing up. And this is extremely worrisome for many of us. Layer on top of this, changes like visas being revoked or denied, federal layoffs, prohibitions on publishing on certain topics, especially for federal scientists. Foreign collaboration is being blocked, and the FBI director has publicly mused about prosecuting people studying climate change with federal grants because he sees that as fraudulent. Meanwhile, policy is being made that could be informed by data, but it's being made ignoring well-established knowledge. There's active movement to delete information from public archives that is policy relevant, and there are policies being made that are being made on the backs of pseudoscience and conspiracy theories. And for people who love understanding how the world works and the truth, this is a disturbing thing. So I want to talk today about how we got to this point and how we can make arguments in defense of our role in society. And so I'm going to start and focus on a question of why does the federal government pay for our curiosity? And for perhaps the older members of the room, I have a gift to inspire this question. We may ask ourselves, how did we get here? And when I gave my practice talk, I realized none of my lab members knew this reference, so I apologize. So let's go way back in time to understand this. So if you go back to the Italian Renaissance or any of the places where science was emerging hundreds of years ago, science was being done by individuals driven by curiosity about the world. And they were able to do that because somebody had wealth and was willing to hand that wealth to them to let them be free to pursue their curiosity, whether that was Da Vinci or Galileo, for example, both of whom relied on the Medici family for financial support. Fast forward to Charles Darwin. Of course, many of you may know that he was rather wealthy. He was a landed gentleman with inherited wealth from the Wedgwood family that made these beautiful plates and bowls. And his father and his grandfather were well-to-do physicians, so he never really had to work. He had the freedom to lounge around at Down House and go for long walks and write letters and travel the world. And so being wealthy helped you do science. Not to say that everybody had to be wealthy. Alfred Russel Wallace was not. In fact, he was quite poor for much of his life, and he made ends meet by traveling the world and collecting specimens that he sold to collectors and to museums. So part of his scientific activity was directly related to income generation. That became formalized by museums, for example. So these four gentlemen were the founders of the journal, The American Naturalist. They worked for Louis Agassiz at Harvard, who had his own batch of money, but they chafed under his direction, which was rather heavy-handed. So they left and went to the Essex Institute, which is now the Peabody Museum in Massachusetts. And the museum had sponsors, and it also charged a fee to enter. And so the museum funded their salaries and funded their collecting expeditions that generated materials to show in the museum. They also supplemented their income by producing a brand new publication, The American Naturalist, which was marketed to a very wide audience. It wasn't initially targeting scientists and academics. It targeted anyone interested in natural history. And it was really quite popular at first and was read by a wide variety of people. And I highly recommend going on JSTOR, going on the AmNat website, and reading some of the early issues of the journal, because they are a hoot. Contemporary with them was Mary Treat, a scientist in New Jersey who published over 80 scientific articles, and she made ends meet by publishing books like this one, Injurious Insects of the Field and Farm, or Farm and Garden, which is a very popular book. It had 5 printings, and it earned her enough income to survive. Then you had professional scientists emerging more in the late 1800s, early 1900s, who had to rely on private foundations. So we were in the robber era, barren of, maybe still are, arguably, but of the late 1800s, and individuals like Carnegie and Rockefeller, who had vast sums of money, as part of their showing off to each other, created foundations to fund these poor scientists working in the margin. And so Thomas Hunt Morgan was funded by the Carnegie Institute. Theodosius Dobzhansky initially was supported by Carnegie and then by the Rockefeller Foundation. And so these were institutionalized sponsorships driven by individual wealth. Now, there's a lot of redundancy and a lot of inefficiency to each individual wealthy person choosing who to fund and who to not fund. And so in Germany in the early 1900s, there was a move to centralize this process. It was still private wealth, but the Emergency Association of German Science was formed in 1920 to take dues from companies and wealthy individuals and create a centralized system for distributing research grants. And an example of this is the following grant proposal by chemist Otto Warburg. The translation here is, I need 10,000 marks. That is the entire grant proposal. I think we need to write to our program officers and suggest some changes. So back to the US and back in time a little bit, the institute that we now know of as the National Institutes of Health began as a single room staffed with one person, Joseph Kenyon. So germ theory was sort of getting big attention in the late 1800s, and it got political attention because we had a big wave of immigration, and there were concerns about immigrants bringing in cholera, yellow fever, and infectious diseases at a time when we finally understood what those diseases actually were and how they worked to some degree. And so Congress supported one room in the Marine Hospital Service, which served sailors and sort of kept health services for sailors, to try and study germs and understand their basis and what we could do about them. And he was the sole employee for 15 years, moved to Washington, D.C. Congress passed an act to build a building, expanded his office in the early 1900s, and then in 1912, Congress created the Public Health Service, which was a predecessor for what we now call the NIH. And they had departments, pathology, zoology, infectious disease, pharmacology. They funded research on things like water pollution and water pollution's effect on human health, air pollution, things that we sort of sympathize and recognize with today. Now, a lot of the activities of the PHS were taken over by the military during World War II and kept very much under wraps, because the military was keenly aware that the majority of soldiers didn't die from bullets or bombs. They died from wounds afterwards or from infectious diseases. And so wound care, antibiotics, and the study of infectious diseases and vaccines were crucially important to the war effort and were managed by a very secretive branch of the Army that ran a massive research organization. FDR, President Roosevelt, was very aware of the importance of research during this time. Everything from radar to, of course, the Manhattan Project to health services were emerging as crucial bits of technology during World War II that gave us an advantage. And so towards the end of the war, he wrote this letter to Vannevar Bush, who was the head of this military Office of Scientific Research and Development. And he basically said, why can't we keep doing this during peacetime? And on the second page of his two-page letter, and you can find this online with a Google search, it's a fun read, it'll take you a couple minutes. On the second page, he says, can an effective program be proposed for discovering and developing scientific talent? So he had in mind not just creating new technologies for war, but also peacetime technology and cultivating scientific talent, and saw that as important. So Vannevar Bush came back a little while later with this report, Science, the Endless Frontier, also well worth your time to read, because it echoes many things that are familiar today. And this led to the origin of the National Science Foundation in the form of the National Science Foundation Act of 1950. And I want to highlight one thing. On the very first page of this act is a passage about supporting scientific and educational activities to appraise the impact of research on industrial development and upon general welfare. So economic benefits and welfare, not in like welfare as we think about the federal program of welfare, but just well-being. I also want to highlight to award scholarships and graduate fellowships. So grad fellowships were on the very first page of the Congressional Act that created the NSF. These are fundamental to NSF's mission right from the very beginning, as is the interchange of scientific information, like conferences, including with foreign countries. So foreign collaboration is, again, on the first page of the Act, a central feature. So NSF funding began quite modest, $225,000 in 1951, slightly bigger in 1952, and that was distributed in the form of 98 grants, so not very big grants. 1952 also was the start of the Graduate Research Fellowship Program that continues today, although it's been somewhat curtailed. And 1957, there was a big uptick during the Red Scare with Sputnik, the Soviets launching Sputnik into space, and the Americans realizing we're behind in science and technology, and that led to a rapid uptick in investment in NSF and other federal research programs, which leads us, after a period of sometimes growth, sometimes stagnation, sometimes slight declines, to the present day, where we have this remarkable and historically unusual phenomenon. We have the privilege of going to the federal government and asking for money to pursue things that we're curious about. That has not been true for most of human history, and it still doesn't make it easy to get the money, but we have that ability to ask. And that's really a remarkable privilege. And it's not a one-way street here. Something is asked of us in return. And this is really what I want to focus on for a lot of my talk today, is what is asked of us? And I would like to think that what's asked of us is simply that we go out and discover something really cool. That we stick our heads out of the bubble of the things we know and understand something fundamental that we don't currently know. And that's why a lot of us are here, because we find that really exciting and enticing and enjoyable. But that is probably not what motivates Congress to hand out money, I regret to say. I think what they really want to see is this. Economists estimate that every dollar invested in basic science by the federal government leads in a 10-year time frame to about a $2.56 economic growth. So about a 250% return on investment over a short time scale, and it's estimated that might be tenfold greater than that over a multi-decadal time scale. So economic estimates of this vary a little bit. This is kind of on the low end. It might be bigger than this, but it's clear that spending money on basic curiosity-driven research is one of the best investments in economic growth that the federal government can make. And it's not something that private companies or individuals will make because the return benefit is not necessarily back to the spender. It's a very generalized and unpredictable return on investment. So what I want to do is a little self-reflection. I'm going to float in a pond for a second and meditate on this. And ask, have we held up our end of this quid pro quo bargain? And specifically, I'm going to self-reflect, and I'm going to ask, have I, as an example? And I think what I'll outline a little bit about my career, I think is pretty typical of a lot of people in this room who have been in this career for a while. And so I'm going to look in the mirror a little bit as an example, as a case study, and ask, are we doing what Congress expects us to be doing? So I started out with NSF funding right at the outset of my career. Actually, my undergraduate research experience was enabled by an NSF grant to the professor who I was working for, Colin Oriens. Then as a grad student at UC Davis, I had an NSF graduate research fellowship. I got an NSF doctoral dissertation improvement grant to help me pay for the cost of my research, and that modest investment led to a handful of papers, including a paper on the ecology of individuals, which is what Becky mentioned in her introduction, which has been my most impactful single paper. And this was co-authored by myself and 6 other PhD students. There were no faculty involved in this. A number of us were NSF graduate research fellowships, and we had the luxury of having the intellectual freedom to sit around a room, sometimes over beers at a bar, sometimes on campus, and just talk until we came up with an idea that we were excited by. Since then, I've been supported by a mixture of sources, and this is a little timeline of my federal funding and private foundation funding. So we have some intermittent NSF funding, sometimes gaps, sometimes a couple of grants at a time. More recently, some NIH funding, and then some private foundation funding in yellow on the right from HHMI, Packard Foundation, and the Moore Foundation. And so total, I've gotten well over $10 million in federal funding in my career, and it's reasonable to ask, what have I done with that? And has that been worth it? And I will let you all be the judge. I sometimes worry about this question. So let's start with discovery, poking our heads out of that bubble to find something new. And this is what motivates me most of all, or at least it did initially. And I started out as a grad student really interested in speciation, and I got very intrigued by sympatric speciation because it was controversial. And there seemed to be a few examples of fish species pairs in lakes that were maybe sympatric speciation, like these benthic and limnetic stickleback species pairs that are found in 6 lakes in the Pacific Northwest on Vancouver Island and nearby islands. But when I started digging into it and reading a lot, I realized that the vast majority of stickleback populations in lakes like this one did not speciate. There's just one population here. They're phenotypically unimodal, genetically well mixed. So why is speciation not happening, and what's going on in these species, single species lakes? So I started looking at phenotypic and ecological variation, and using a combination of diet analysis on the right, stable isotope analyses on the left, we can document massive between individual differences in diet inside a single lake. So for example, on the right, each black dot is an individual fish. They're connected if their diets are very similar to each other, and we see these dietary clusters, individuals, a group of individuals who are all eating benthic cladocera, a group who are all eating chironomids, a group who are all eating pelagic cladocera. And that diet heterogeneity is persistent, and so it's reflected in stable isotope data on the left. Each little dot is an individual from a single lake, and the boxes, the rectangles, are this averages for the limnetic and benthic species pairs. So you can see the individual to individual differences in diet in a single lake are as large or larger than the species differences in those famous species pair lakes. And yet, the morphology in these lakes is unimodal, not bimodal. And so individuals at one tail of this trait distribution will tend to eat one kind of prey, and individuals at the other end of the distribution will tend to eat other prey. And so I had some papers about that early in my career, this individual diet variation, and that it turns out is being sustained and inflated by resource competition, generating disruptive selection, and my PhD work was documenting that competition inflates this disruptive selection. Early faculty job showing this disruptive selection is really common in a lot of lakes, but it's more common in certain kinds of lakes than others. But in the process of doing that, I got distracted by tapeworms, as one does. So this tapeworm shown here in the photo is massive, right? These can weigh up to half the body mass of the fish. And you can't help but notice them, but it took me a while to notice them, right? I'd see them, I'd note them down, and then finally I started paying attention and realized that, okay, what are these things? What's their natural history? And it turns out their natural history is they're transmitted when the fish eats an infected copepod, which is infected from eggs. Then birds eat the fish, the tapeworms mate in the birds. Now this matters here because individual fish who eat copepods are going to be more likely to get this parasite. So the diet variation I was studying is tied into infection risk. Only some individuals and primarily some populations are going to be heavily exposed, whereas the more benthic individuals or populations are going to not even drink the Kool-Aid. And sure enough, there's massive between lake as well as between individual variation and risk. Each dot here is a different lake on Vancouver Island in British Columbia, and we go from lakes with 0 tapeworms out of hundreds of fish sampled to populations where 60, 70, 80% of the fish are infected. And I've seen lakes in Alaska with 100% infection rates. So I started asking, why is this parasite prevalence so variable? And to cut to the chase, ecology, diet explains a little bit of this, but there's a lot of residual variation. So I got interested in whether immunology could give me a toolkit to understand species interactions at a mechanistic and genetic level. So I started doing flow cytometry and ELISAs and histology. I hired an immunologist, Natalie Steinel, as a postdoc. She was a mouse B-cell and mouse T-cell immunologist, and she was brave enough to launch into Stickleback evolutionary ecology. And she was in the lab doing injection experiments, putting fluorescently labeled artificial antigens into fish to trace how macrophages were carrying those antigens to immune centers in the spleen. And she knocked on my office door one day and said, "Hey, Dan, I've been dissecting a fish, and I see something really weird. Can I show this to you?" And I apologize for the following, but I'm going to show you a video of a dissected fish because it's important to the story here. Just don't get sushi later today. So on the left is a normal stickleback. You can see the body cavity is splayed. That's the liver being shown to you, and it moves around just fine. It's loosely, barely connected to the intestine underneath it just by some vasculature. And on the right, you see these thread-like structures, and we can't even open the body cavity without ripping it because all of the organs are glued together. They're glued to the body wall by these thick fibrotic threads. And this is a scar tissue of extracellular matrix, fibrinogen and collagen. So what Natalie showed me, I said, yeah, I've seen that before. So it turns out this was being induced by her injections, but I'd seen this in the wild for many years and ignored it because I just thought maybe I didn't preserve the fish well enough. I didn't flash freeze it enough, or I didn't get the formalin in it as fast. And I just thought it was something spurious, a mistake on my part. But it turns out that it's fibrosis, which is a web of extracellular matrix laid down by fibroblasts, collagen and fibrinogen. It's scar tissue, which is a common inflammatory side effect. And it turns out that in stickleback, there are populations with a lot of fibrosis, populations with little fibrosis, and in F2 hybrids between these populations, we see that individual fish that are genetically capable of a fibrosis response have smaller tapeworms than individuals without fibrosis in laboratory experimental infections. And when I say smaller, I mean much smaller. This photo on the left here are tapeworms that are all 42 days old, and these tapeworms are full siblings of each other. The difference is the fish genotype and whether the fish had fibrosis. So that's a massive effect, and when a fish has tiny tapeworms and fibrosis like that, they can encase the tapeworm in a cyst and kill the tapeworm. So it is an effective immune response that can eliminate the parasite. Unfortunately, that fibrosis sticks around for a long time afterwards, which is unfortunate because it's costly. This fibrosis changes fish's body stiffness, which can alter their escape response from predation. It reduces their fecundity, it increases metabolic costs, it decreases their foraging success. So this is a really harmful immune phenotype, although it's an effective defense. And it's harmful not just in stickleback, but it's harmful in humans. So approximately 30% of deaths worldwide involve diseases that have fibrosis as a crucial element in the organ failure. This includes a lot of cancers, heart disease, pulmonary fibrosis, cirrhosis of the liver, scleroderma. There's a large number of diseases where fibrosis is a major component of the harm and degradation of body function. So what's cool is we now have data from a large number of lakes on Vancouver Island, and there's substantial variation in fibrosis severity between populations. Some populations have really intense fibrosis, some have very little. And when we take these fish into the laboratory as eggs for common garden rearing, we see variation in baseline fibrosis. Some lakes have severe fibrosis even if we don't do anything to them, and others only when we challenge them with an immune response, feed them a tapeworm, inject tapeworm protein, or inject an immune adjuvant like alum. And so we can do tapeworm protein injections and induce these immune responses, and the magnitude of that response varies between lakes, and not just the magnitude, but the speed of onset and whether it's reversible. There are some lakes with fast onset, but then they can recover, which is cool because humans, once we get severe scarring, it doesn't go away. But in some stickleback populations, they can actually recover and greatly ameliorate and reduce their fibrosis lesions. But other stickleback populations cannot, which means we have an opportunity to study the genetic basis of variation in fibrosis onset, severity, and recovery, and have a new model organism for studying fibrotic diseases. So we've been doing genetic mapping of fibrosis, combination of QTL mapping, population genomics and selection scans, identified a gene SPI1B in our QTL for fibrosis that was the strongest target of selection. This gene is a transcriptional regulator of fibrosis in fibroblasts in mice, so it's a pretty good candidate. We did CRISPR knockouts of SPI1B and confirmed that it modifies the fibrosis phenotype in stickleback. And then in humans, there is a drug in development called DB1976 that targets the protein product of SPI1B and inhibits its ability to bind to its own target. And if we co-inject an immune stimulant that causes fibrosis with varying concentrations of this drug, we can knock down fibrosis in stickleback, confirming that the protein product of SPI1B is directly involved in activating fibrosis. So stickleback genotype matters for fibrosis. It turns out so does tapeworm genotype. Different tapeworm populations vary in their proteome composition, and we can inject purified proteomes into stickleback, and we find that different tapeworm populations induce different levels of fibrosis in a particular host genotype. And we're currently working on the genetic mapping of the parasite side of the story. So to wrap up my brief research bit, I couldn't leave it out. Curiosity-driven research, I started out interested in sympatric speciation, for crying out loud. Was enabled by NSF for some reason, and it revealed by accident, unexpectedly, natural genetic variation in fibrosis susceptibility and recovery that's produced a new model organism for studying a human disease. We don't have good natural genetic variation models of fibrosis outside of stickleback. So this is really exciting, and we've had NIH and NSF funding both now, the NIH funding is really building off of this story. So if I go back to my meditative state for a moment, I feel pretty happy, but I will acknowledge I've not saved any species from extinction. I've not solved climate change. I've not produced any patents. I've not healed anybody from any disease. So I would say that this is very much a work in progress. My hope is that sometime after I retire, somebody builds on the work that I've done and maybe produces a drug inspired by tapeworm immune suppression strategies that might help reduce fibrosis, or based on what we find about how fibrosis can be reduced and recovered from in Stickleback. So I would say I've not really generated a whole lot of direct benefit from my research. It's been interesting. I enjoy it. I hope you find it interesting. But it's not going to be something that I wave in front of my congressional representative and say, look, I discovered this thing. But what I have done that I think is actually much more important is training. Remember, Roosevelt's letter to Vannevar Bush highlighted training as a core goal. The NSF Act of 1950 listed training on its very first page. And I've done training. I've had 29 postdocs. 24 of them are faculty, one's a doctor, one's a fishery researcher, and 3 are still postdocs currently with me. And so that's 83 person years of employment made possible by research funding, mostly federal. I've had 26 PhD students and a smattering of master's students. Some of them have gone on to be faculty, but many of them are teachers, many of them are in startup companies, many of them are working for larger industries or nonprofits, and I feel really good about that. I don't think our sole goal here is to produce more people like ourselves. We should be creating people with scientific expertise who go out into the broader workforce. That's one of our purposes, one of the goals of the federal funding. And that's especially true with undergraduates. I've had 150 plus undergrads come through my lab in 20 years or more as a faculty member. And some of them have gone on to be physicians, actually quite a few. Some have gone on to be epidemiologists. Some have gone on to be scientists. Some of them may be in this room today. And I'm super proud of all of their accomplishments made possible by federal funding for the research they did and the salaries they received. Science teachers from high school and middle school have worked in my lab for summer research experiences and in the field with me. There's strong evidence that when teachers have research experiences, it strengthens their capacity to train their own students in their classrooms. And then that training is not just about developing talent within the US, it's also about attracting talent into the US. And I'll highlight one of my former graduate students, Kelsey Zhang. She came from Beijing to do a PhD with me. She was funded by NSF money. And after her PhD, she went to be a data scientist, worked first with Airbnb. She helped found Airbnb Experiences. She currently works at Meta. She's one of the top data scientists at Meta, studying how Gen AI can be insulated from our collective biases and how AI can be made safe and equitable. Unfortunately, that work is being curtailed by Jeff Zuckerberg right now, but I'm super proud that somebody who learned to love statistics in my lab has gone on to do something very productive outside of academia. And I think that's the kind of thing that we ought to be trumpeting and announcing widely. Science can also guide policy. I'm not a climate change researcher. I often kick myself for that. I feel I ought to be. But I have had students do policy-relevant work. And I'll highlight another student from outside of the US who came in, Sebastian Stockmeyer, who's now professor at the University of Tennessee in Knoxville. And Sebastian came in to study immunology and behavior in bats, vampire bats in particular. And he could inject bats with LPS and induce symptoms of illness. And he's found that the social networks of vampire bats were modified when they perceived each other to be ill. And that led to a review paper that we wrote in 2019 and set aside. And then 2020, we dusted it off and published it about infectious diseases and social distancing in nature. How do wild organisms evolve social distancing strategies to handle infection? And that turned out to be pertinent to, I don't know, something that happened a few years ago. And I don't know specifically of any politician who read this or invoked this, but I'd like to think that over time this could build into a wild organism, evolution-guided set of principles for what works in social distancing that might inform policy in the future. Now, economic impact is a tricky one because I haven't patented anything, I've not developed any product, and this is fundamental to Roosevelt's instructions and to the NSF Act. I have had an economic impact in the form of employment. All of those grad students, postdocs, undergrads, they get employed and that money goes into the local community. There are research technicians, like the individuals shown here, who I've employed. They pay rent, they go to restaurants, they boost the local economy, and federal research dollars echo through local communities, not just being spent once, but being spent repeatedly. But there's also economic impact of other forms. And this is an unexpected one. And I'm going to tell you a quick story about my current PhD student who's graduating this year, Swapna Subramanian. And Swapna came to my lab to study parallel evolution and evolutionary prediction. And she was going to work on lakes in Alaska. We have a big experiment where we founded 9 whole lake populations of stickleback. And she was going to work on that. But she got an NSF grant, a graduate research fellowship, which gave her the freedom to do whatever the heck she wanted to. So one of the things in central Connecticut that's a lot of fun is there are lots of apple orchards. We're obsessed with apples in the fall. And Swapna realized these apples have yeasts. And there are many orchards. Every orchard has different varieties like Honeycrisp and Golden Delicious and Macoun, and then you go 10 miles away to another orchard and you again have these same varieties. So she got interested in predictability and repeatability of yeast adaptation to apple varieties in replicate orchards. So she did big yeast experimental transplants between apples, did population genomics of the yeasts, and that's all work in progress, either in review or just about to be submitted. But along the way, she met apple orchard owners. And one of them was a young man, Jeff Rogers, who had come back to his family farm, 8th-generation family farm, and was starting up a cider house operation, Longview Cider House, which I highly recommend if you go through Connecticut. And Swapna and Jeff started talking and realized that the wild yeast she was purifying and studying might be useful. So they worked together for a couple years. They got a small business grant from the state of Connecticut. And about a month ago, Swapna and Jeff and I and another student, Emma, who's going to be here later today, spent a day crushing 3,000 pounds of apples to create our first commercial run of a wild yeast, purified wild yeast strain that yields an absolutely spectacular cider. And so this is the kind of thing that when I went to Washington, D.C. a few months ago, I did wave in front of my congressional representatives. This is a graduate student research fellowship enabling a town gown collaboration, helping a family farm startup company produce a new product. Now, I study Stickleback, right? This is not my work. This is Swapna's work in its entirety. But federal research grant and my lab existing made unexpected side effects happen, which is super cool. And so I feel pretty good about myself. I've trained, I've produced policy-relevant work, I've had students produce economic impact, and then I've discovered things that I think are pretty neat. And so I'm going to challenge you to do a little bit of introspection and ask, are we collectively doing what we ought to be doing to merit the federal investment that we receive? Because I will tell you, many Americans don't see the value of what we do. This cartoon, I think, exemplifies an attitude that is not uncommon, even in the halls of Congress, that we might be in what might be called the Department of Trivial Nonsense. And, I looked back this morning at some of my research paper titles, and they include things like a spectral framework to map QTLs affecting joint differential networks of gene co-expression. I hardly know what that means. I mean, I do, but my family doesn't, my neighbors won't. And so it's no surprise that people see this as a department of trivial nonsense. And so some people are actively hostile to what we do as evolutionary biologists. This is the 100th anniversary of the Scopes trial. But some of that public skepticism is because we're doing things and we're not communicating it well, and that public skepticism is undercutting political support so that research grants may become a mythical being. And so I think with a obligatory XKCD cartoon, since Jeremy had one yesterday, I had to add one too, we need to do better at conveying the value of federal funding for our curiosity. So what is it that we can do? What can we do as societies? What can we do as individuals? So I did get a letter earlier this year saying scientific society should stay away from politics as much as possible. And I think there are some reasons for that, but also some good counter-arguments. So this is the Constitution of the American Society of Naturalists. As a nonprofit, we have to have one. It's filed with the IRS. And it says our objective is advancing and diffusing knowledge. But it also says that no substantial part of the activities shall carry on propaganda, attempting to influence legislation, participating or intervening in any political campaign, including publication and distribution of statements. So there are some constraints on what we can do. But diffusing knowledge. Diffusing knowledge isn't just to each other. It's not just in scientific articles. We need to inform the public and policymakers about what we do and about issues for which we have direct expertise, whether that be climate change, epidemiology, conservation, evolutionary biology. And then advancing knowledge requires political items like funding, supporting a diverse scientific workforce, free speech, free inquiry, free association, equitable access to scientific knowledge. And so I think societies are obliged to make statements publicly about the value of what we do, inform policymakers and the public about scientific principles. And so we can, as a society, speak for our membership at large by writing letters, by co-signing letters, and by going to Congress. And this is a photo of myself, and then third in the line is Katie Lauderhouse, ASN secretary. We were on Capitol Hill back in March, meeting with legislators, or maybe it was April, meeting with legislators to talk about the importance of scientific funding and what we do collectively. And this was part of a American Institute of Biological Sciences. We were there as ASN representatives, and it was pretty cool to walk into offices of one of our representatives, walk into Elizabeth Warren's office, and the staffer says, okay, it's the American Society of Naturalists coming in, and I'm like, yeah, that's right, it is. We also can be involved potentially in legal actions. This is one of our best recourses to fight what's happening in Washington, D.C. right now. Several of the societies here were contacted by a union representing federal workers asking us to join a lawsuit on behalf of NSF program officers, and we were within a hair's breadth of being co-plaintiffs on a lawsuit against the Office of Personnel Management on behalf of NSF program officers. The officers in question were rehired and we lost standing to do so, so it became moot. But we have the ability to do this. It is a risky thing for societies to file lawsuits. We do not have deep pockets. And so this is something that we need to do cautiously, but I think we have the ability to do and we need to take very seriously. We can also encourage you all, provide members with information and inspiration and motivation to take effective action. So you all have the ability to act in some ways that we don't. As society president, I cannot formally lobby for legislation, but you as individuals can. So you can carry out science communication. We need to tell our public, we need to tell voters why we do what we do and why it's important and effective. And so that means op-eds, letters, long-form stories, videos, blogs, vlogs, etc., aimed at the broad public. Tell personal stories with emotional resonance that connect with the audience. Choose your audience well. News outlets that are speaking to the choir are not going to change hearts and minds of voters who need to change what they do. We need to get Republican voters to call their Republican representatives to change their minds about scientific funding. Yeah, it's tricky. You can contact your representatives. Calling is best. Emails can be automated, and so they have less weight. So calling, you can also set up a meeting with your representative or their staffers in your home state or if you happen to be in Washington, D.C. You can educate policymakers. You can go and give them information. One of the biggest surprises for me when I was in Washington, D.C. was talking to staffers, and they said, we don't know what's happening at NSF or NIH because the White House directed a lot of federal staff to not talk to Congress, and in particular, not talk to Democratic representatives. And so a lot of our representatives who are crucial for funding decisions are left in the dark about what's happening within the executive branch agencies. You know when grants are being canceled, you know when programs are being canceled, but your representatives may be unaware of this. So let them know, inform them, give them the information that they need to make appropriate action. You can help your representatives. You can write reports that give them information and update them. You can also suggest questions to ask in hearings. There will be a nominee for a new NSF director because the recent NSF director is stepping down. You can give our representatives suggestions for hard questions to ask whoever is nominated. You can stay informed. It's tempting to hide our heads in the sand and feel very depressed, but we need to be active right now. This is not a time to say, it's too depressing, I can't hack it. We really have to be raising our voices. And then we can do the thing that comes naturally. We can continue to do the things that the federal government is not doing. When the federal government is not supporting diversity, equity, and inclusion in science, we as individuals, as departments, as societies, can continue to do so. We can also keep doing good science. This is a time when people in power are ignoring facts and ignoring data and information, so the pursuit of knowledge is, in and of itself, an act of resistance. And so while I know that during this conference this week, there will be a lot of hallway conversation about how hard the current political situation is and how worried we all are, I also want you to revel in how cool the science is that's being done, how impactful it may be, and how interesting it is, and I want you to enjoy that science because that is why we are here, and that's extremely important. Thank you all for coming. Thank you all for acting. Thank you all for being members of these societies, and enjoy the conference, please. I believe I have time for a couple of questions. The official time ends at like 9:15, is that right? And I can't see anyone with the, oh, there you go, now I can see if there are any questions. Looks like everybody just needs their coffee right now. Okay, yes.
Differentially the ones which are addressing questions such as diversity, equity, and inclusion. For example, my NIH-funded genomics, data science, cloud computing training center for minority-serving institutions will be terminated on June 30th. We were told by the NIH that our work no longer met NIH priorities. Now, who in this audience thinks that genomics, data science, and cloud computing is no longer an NIH priority? What they were really saying is we don't want black people to be trained in these tools. And so if you look at these terminations that are occurring, they are occurring for researchers at institutions like mine, historically black institutions, minority-serving institutions, Hispanic-serving institutions. They're also being terminated for their topics, such as, gender biology. And so it's not uniform across the board. So I'm glad that you mentioned DEI at the very end of your talk. Of course, if I were giving this talk, I would have started with that. But in addition, folks need to be aware of what the consequences are. So you're in Connecticut. You can individually continue to do DEI work in Connecticut. You cannot do that down here. You will be terminated from your position if you try to do that down here. So folks need to be strategic. Okay, but thank you for your talk.
And even more, thank you for the work that you're doing and the work that you have been doing on getting minorities into data science and genomics and genetics. I'm not surprised, of course, by this grant termination. I've heard many examples of this. I should add that the American Society of Naturalists is, along with other societies, is leading a survey that has IRB approval. Members of the audience who are members of these societies should have gotten emails about this survey. I really encourage people to complete the survey. Katie Lauterhaus, the ASN secretary, has been spearheading this to collect concrete examples of stories like this so that we can bring these up to representatives, bring this to Congress, and create pressure based on data-driven information about impacts of these federal cuts. And yes, you're exactly right that the cuts that are happening are absolutely discriminatory and biased, and it's horrific. It is absolutely horrific. And as you say, one of the things, some of us are in a more privileged position in terms of geography and the politics of where we are to continue doing this and continue doing DEI work, and I think it behooves us even more now to fill the void as much as we can. And I think one of the things the societies are having conversations of, the leadership of societies, is what can we do right now in this time? We have some modest resources that we put towards student travel and student research grants and this and that. Are there ways in which we ought to be pivoting our investment of our limited funds to try and maximize impact given the changing federal support at this point in time? And I think DEI activities are one of the places where there's a vacuum that we have an opportunity to do a tiny bit to fill. It's not enough, of course. We can't make up for the grants that are being canceled.
Hi, thank you for your talk. I think I needed that. And thank you also for the question prior. My question is regarding your talk centered a lot on the economic impacts of our work, and one thing I've been wrestling with a lot and thinking about is the fact that community building, mutual aid, and things like that, these are all, I like to say that progress happens at the speed of trust, and one thing that I find challenging is investment in this work, being that a lot of that work is very intangible. I may invest a lot of time and energy and effort into community, like kind of communicating my science outside of the university, but that doesn't equal dollar signs, or I don't have concrete values that people who are very data-oriented or results-oriented may see. So how do we show the value of the work in things that are quite intangible at times?
So, yeah, I'm not enough of an economist myself to say, here's how we can put numbers on that to communicate it to the people who are often very data and number driven in their policy decision-making approaches. It's a great question.
So I, and I'm certain many of the rest of us in this room, have several science skeptic, MAGA hat wearing relatives and probably close friends. So I was wondering if I could solicit your thoughts in how best can we talk about our work to them, the people who are close to us but who stand in opposition to many of the things we do, and maybe start to change some minds.
Yeah, that's a great question. And that's really kind of broad science communication question, whether it be talking to family, neighbors, friends, or writing op-eds, and a lot of it comes down to starting off with, and there are people in the audience better suited than I to speak to science communication principles, but from what I've learned, a lot of it comes down to starting with common ground, establishing common ground. So for example, maybe you have a family member who loves hunting. Okay, I'm not a hunter myself, but I love the outdoors, I love being in the outdoors. They do too, for a different reason, but there's that common denominator that we can build on and identify what is a shared value, and then communicate how something that we do contributes to that shared value. They don't want to be going hunting in a clear-cut logging area or a parking lot. I don't want to go hiking or rock climbing in a clear-cut or parking lot. There's a shared denominator there. And so a lot of it comes down to trying to identify who's your target audience, what is it that you share with them that you can identify as a shared value, and then talk about how the work that we do contributes to that shared value. And I'll thank the AIBS, American Institute for Biological Sciences. They do this Congressional Visits Day, but there's a 2-day training workshop in advance of that that's all about science communication generally and about communicating science with representatives and policymakers, the budgeting process. The SSE actually subsidizes students and postdocs to go to this. There were a number of SSE members, students, and postdocs who were at that event. It was great. I really recommend it. I think there's another one coming up this summer. So a big shout out to AIBS for that.
Thanks, Dan, for focusing on student training as well, because I think that's a really important part of things, and it's sort of how I help to feel better about myself doing curiosity-driven research is through the training component. But one of the struggles that you made me realize in your talk is that really the societal benefits come when we spin people off outside of academia. And yet there persists this notion that our primary goal and where we really see value is when we train people to do the jobs that we're currently doing within academia. And it's sometimes viewed as a leak from the system when people go off and do something other than academia. And yet, from what you were saying, that is where our societal value really comes, as opposed to just feeding the machine that is academia once again. So how would you suggest we go about changing that perception of what success in training looks like within academia?
Yeah, hear, hear. I will say that I have mixed feelings about this issue, because I actually feel like most of my colleagues, both at Connecticut, where I am now, and when I was at the University of Texas, when the faculty were sitting around chatting, I think most of us were actually pretty supportive of spinning people off, so to speak, into non-academic jobs. I think we all felt pretty good about that. So I think there's a perception that we're less supportive of it than we really are. I think there's more appreciation for contributing science-trained individuals to the broader community than we think. So I think we tell ourselves that we don't support it when we really do somehow. That said, a mea culpa. I highlighted my student Swapna Subramanian. She's brilliant. She's going to be up here giving this talk in 20 years probably. And selfishly, I want her to stay in academia simply because I'll enjoy seeing her at conferences. If she goes off into nonprofit or NGO or government work or company work, or she founds her own cider company, which is possible, I just won't see her as often. So there's that. I'd love to see her stay in science because she would be so good for all of us to be here. And yet, really, if she goes off and founds a really good cider company and makes good money, that's great for her. That was really awesome. I'll be sort of personally a little sad because I won't see her as often. That's it. So I think, there is this tension, a little bit of kind of personal, like, oh, I'll miss them. And yet, the students who I have who have gone off out of academia and established interesting jobs, I think they're amazing. I think it's great. And so how do we change the perception? I think we change the perception by standing up here on the stage and saying, it's great when people go and take their academic skills and apply them in other contexts. That's fantastic. That's what we ought to be doing. All right, I think, let me check the time. Yeah, we should get going. Thank you all for your attention. Enjoy the conference.